pleased to move to the top first session today. As I said, this is going to be Nikola Vlahov from the University of Oxford, who will present his result on a novel regulation of SRC feminine by isoforms.
Okay, right. So first of all, sorry I was late this morning. Hi, I'm going to talk to you about the novel regulation of Stark family mechanisms by F1 isoforms, which I'm not ever going to mention again in the talk. What I'm going to talk about is what is cancer. I'm basically doing deep field in radiation oncology, and I'm just going to tell you what cancer is. Basically, you have a nice body, you have a nice organ, everything's working fine, functioning correctly, you have lots of cells making this organ, but then eventually, with time, mutations occur, changes in the DNA occur, and you get stuff like that, you get breaks, you get mutations, and eventually you get this malignant formation forming. It then asks for nutrition, so it gets blood vessels and becomes a big tumor. And then it releases one of its friends to go somewhere else, which is called metastasis, and it goes to a different site of your body, and unfortunately that's what kills you most of the time, the metastasis. And unfortunately, 1 in 3 to 1 in 4 people in the world will actually get cancer in their lifetime. Doesn't mean that they'll die from it, but just they'll get it. Right, and in terms of, right, okay. This graph here is for the incidence of cancer in Bulgaria. This is the incidence of cancer in the UK. You have the top here is breast cancer because it's combined for both sexes. Then you have lung and so large bowel and lung for Bulgarian population, and prostate and large bowel for the UK. Basically, my point here is that okay, they vary in terms of where you are, what your diet is, what you're doing with your life, but then the actual incidence is quite high. You would get something eventually, unfortunately, and you have to find a way to treat it, or you find a way to find it as soon as possible and to get rid of it before bad things happen. But the way I'm looking at it is using through the molecular biology. I'm basically watching cells and nucleuses and chromosome, genes and proteins, and I'm sure you all remember from your high school that basically you have the DNA, and then you get these instructions that are transcribed into RNA. The RNA is the messenger that carries that message, and you get this translated into a protein, which actually does the work. Now, most people, when they talk about protein, they think about this. I occasionally think about this. But in science, it's actually this. Or if you want, it's this. And this is basically separated on size, different proteins, and you get to use this fancy kit, which is really fun. It's going to be boring. But another type of visualization of the protein is this. I really rather enjoy this than the black and white thing. Our group works on signaling pathways. Basically, we try to fill. Oh, right, okay. There are things written here. So they lead to one to the other. It's basically a cascade of events in the cell that leads to different responses, to different signals. Okay. Right. This is very populated slide here with all the metabolic pathways in the in the cell, which you can see, but it's very populated, it's very dense and complicated.
You can maybe turn the screen towards us. Computer screen.
I'll show you later. But these are all the pathways that, or some of the pathways that are going on in a cancer cell, in a regular cancer cell. And only a few of them actually. Now, what I'm doing on an everyday basis, I'm dealing with this F1 gene. It has 2 different isoforms, 2 different forms, a good form and a bad form. And we basically believe that the good form keeps the bad form at bay, but when the good form is lost in cancer, you get the bad form doing whatever it wants. Basically, it's this gene, the good one, is inactivated in many, many cancers, which you cannot see, but trust me. And then basically, if we manage to treat that, if we find a way to use a drug or reverse the process or do something else, we get to treat all of these and all of these. not to cure them, but at least to help them, to get a better prognosis, to get a better outcome, and so on. So, what I usually do, I put the two proteins inside cells, so you have the good one in red, the bad one in green, and eventually you see that yes, okay, it's actually the good one is keeping the bad one at bay. So the more the red is expressed, the more the green is lost. And if you want to use a simple microscope, you get to see something like that. Basically, these are just normal cells which have this overexpressed green protein, which is at this junction, which is basically holding the epithelial cells together, which means that if I poke my finger to my hand, my finger doesn't go through. It's what keeps the cells tightened together. And when I saw it, I hope it plays. Okay, interesting. No? Right, sorry about that. In a moment. Okay. Anyway, the whole idea is that they are moving constantly at a constant rate. It was very beautiful. Again, trust me. It was okay. Basically, it looked like that. Basically, you had green things flowing around. It looked like the airplanes in the sky of the world. And I thought it's one of the most beautiful things I've ever seen in my life. The microscope, it looked like that. And here you have the example of the good and the bad one. So the good one is, constantly moving. Things are flowing around, transporting stuff. But then the bad one is actually accumulating these big blocks of green. And we wanted to investigate what's going on. Why is that? So we wanted to count all these things. Now, it's not that easy to count them one by one. So we use a software which basically told us, okay, so they're going that way or that way. Told us the direction. Told us the actual distance they've traveled, told us the speed, and eventually we wanted to assess the actual junction, so where they're actually holding together, and we used the CRUK technique, which is called fluorescent recovery after photo bleach. We basically use laser to bleach the area and see how fast the molecules actually return, and this tells us how tight the molecules are kept and gives us all kinds of information about the structure of the two cells. And then if you do many other experiments, you get to the point where you do all the cool experiments, which is this. Basically, you put the good and the bad cells in gel, and you get to see that the bad ones actually become these nasty, massive things, which is more invasive. It proves our theory. Or if you want, you can actually inject it into the brain of a mouse, and you get to see that they actually grow bigger tumors. And you get to see that if you put cells, so this is the bad protein, so this is the green you saw just before, you get to see that as the cells move around, you actually release this trail of stuff behind. And also, if you have this gel growth thing, very invasive, very bad, you get to see that only one cell is there. And we thought, okay, that's kind of interesting. We also saw that this green cell, if it works again, it doesn't, okay. This pops out and just stays round, trust me. This basically we thought that, okay, it's this one cell that's actually communicating with all the other cells and it's telling them what to do. So they eventually become kind of green, so that's bad. And they become more invasive, which is also bad. And which led us to exosomes. So exosomes are the theory behind this, that basically it's telling the cells somehow what to do. Exosomes are nanoparticles, they're around 100 nanometers. Very, very small. They're secreted from the cells. How you get them? You basically grow cells. You get the red thing, put it into titanium tubes in titanium rotor, put it into this big centrifuge, and you spin it only for 18 hours at 100,000 times the acceleration of Earth. You follow this protocol, very simple stuff. But eventually you can actually get them, purify them, stain them, and put them on cells, and you can actually visualize them. And you see where they're going, what they're doing. And the coolest thing about what they could be doing is that they might be actually promoting this invasive phenotype that we saw before. So I actually did that. I took normal cells, put them with these exosomes, and we got to see that the bad exosomes actually get this bad outcome. And that's what we're, okay. that's what we're doing in the signaling pathway laboratory. Basically, we go through different parts of the cell. We try to investigate what leads to the next, what leads to the next thing, how we can answer this question, how we can fill these gaps, which then tell us, okay, if we have these things and we know these things, then we can use one drug or the other drug or a combination of drugs to actually treat these patients who actually have that particular mutation, that particular disease, and so on. And you get to filling gaps like that, which on here there are no gaps. But basically, you get to fill in all these missing parts of what's going on. Unfortunately, even after so many years, I've only managed to assess only this small part of the pathway. The rest actually remains. But having said that, whenever a patient comes up with some messed up part here, but not so much here, you can actually give him something. It can actually make him better and more successfully. And if you use a chocolate to make a model, basically you can have the green cells secreting all kinds of stuff, making the cells round, making the cells more invasive, and moving places and basically getting metastasis. Thank you for your attention.
Any questions?
Hi, thank you for a really good talk. So this protein sounds like an RAS-related one. Is it
Yes.
again an oncogene and then
Yes.
a follow-up to it? The good and the bad forms are different folding of the same protein, or
They're
they're
the different isoforms of the same protein.
oh,
So one is a tumor suppressor, the other one is well, potential oncogene. We are currently trying to prove the world that it's oncogene.
it's the same protein,
It's the different isoforms of the same gene.
the same. So
Yes.
I don't tell you this one.
Yes.
So what does this protein actually do?
Oh, that's what we're trying to find out. Basically, what it does is it makes the cells want to go places. And it facilitates that. So basically, if I'm holding hands with somebody and I want to go that way, I can because I'm holding hands. But if I'm not holding hands, I actually release this. I get this signal in the cell to actually release this person. I can actually go wherever I want, which is basically what happened with cancer. So usually cancer, especially in the epithelial layer like your skin or whatever, it actually hold together quite tightly. But then they get this signal to, oh yeah, I don't want to be next to that thing. I want to go somewhere else, and then you get this massive formation and you get metastasis. So that's one of the things we actually examine, that it does this. Or it secretes these exosomes things, which are kind of facilitating the metastasis when it gets there. So you have the release signal, but also you have the travel, and sometimes the cells don't make it. Technically, only 5% of all the cells that can be released from a tumor can actually get to the site of distant metastasis. Yet these cells are the ones who would actually kill you, and then somehow they find it better in that particular area. Or, for example, we have different cancers gives you most of the time metastasis in different, actually specific places instead of just randomness. Oh yeah, let's go there or whatever. So we're actually trying to examine how our cell of interest is actually facilitating this process, and how it's telling that other place in the other part of the body, oh yeah, we should probably accommodate this cancer cell to kill us. That's simplified.
So it probably depends on the extracellular matrix, how it's attached to it.
Yes, one of the things, yes, it is. I mean, the exosomes are okay. The exosome would not change that, but they change the actual response. For example, how the epithelial cells of the veins are more adhesive, whether they will allow the cell to actually go there and penetrate, and so on. So yeah, the matrix is involved, but not particularly in this process.
Thank you.
You mentioned that there's a potential drug for this.
Yeah, there are two drugs that could be used. One is the two of them are inhibitors. Basically, you can use them in particular cases. They are already in the clinic, but the case is, okay, you have this mutation in your cancer, whatever, and whenever we know that, we can actually treat it with a variety of drugs and try to kill it as fast as possible from different angles, instead of the normal theory, which has been for lots and lots of years, which is, okay, you have one thing, we give you one drug, oh it doesn't work, we give you the next drug, oh it doesn't work, and so on. You basically try one at a time. No, the recent theories, first of all, your tumor is heterogeneous, basically all kinds of mutations occur every day in it, and then you want to kill everything with a cocktail of drugs. And that's what we're trying to see, okay, to prove to the basically medical community, if you want, okay, if you have this mutation, if you have this problem, you need to probably give them these drugs because they will benefit more. But to do that, we need to prove what we're doing instead of, just making a theory.
So it hasn't reached the clinical trial process?
All the drugs are in use now. Just the combination of them is a matter of proving, okay, you should probably do that. I mean, it's like a suggestion, but unless you base it on facts, you can't really tell medics, oh yeah, why don't you, try that thing.
I was also going to ask about the drugs. Do you develop them, or do you work with pharmacologists who make
No, we
them?
don't develop them. Basically, they're available. You can buy them from any supplier for now to use them in vitro, like our system. You can get them easily.
So they already exist?
Yeah, they're already existing. Yeah, they're already treating patients, different patients, but the point is that particular mutation can give you better response if you use both of them or more, for example.
Have you mapped the mutations that will give you the so-called good and bad ones?
It's not mutation, it's methylation. So it's
genetic.
epigenetic process. And it's hypermethylated in most of human cancers.
And do you think that the exosomes are involved because they might be transporting the protein itself somewhere, or it's just somewhere, something that is equal to
I think they're transporting the mRNA of the bad one, but what does this do? I probably need another 4 or 5 years to do that. To assess. But yes, that's what we do. And just to make one point, usually, in especially Bulgarian media and so on, you get this big stories about, cancer drugs that are constantly being discovered and so on. Well, unless they actually go through, a whole process of have they actually cured that many people successfully the right way, this all kinds of assessing of the whole situation. It probably, just people trying to get, newspapers sold instead of just trying to help people, because they give them false hope and then it actually that crushes at some point. So yeah, it's something that really saddens me every time I get to see it in the newspaper. Oh yeah, we always had this drug, but we didn't want to give it to people. Now we decided to give it to people, and suddenly, oh yeah, please buy it, and it actually doesn't work.
But you can be the next Bulgarian to cure cancer out of many.
Yeah, but I will get to prove it. That's the cool part. And then thanks for your attention.